Hi everybody, it's Sunday, which I count as the end of the week because why not? Running a job, and it will be done in an hour and 11 minutes, so that's how long I have to record and edit this before I'm back to lab. So yep, let's go. So as you may recall, back in September or October, NASA had a really big press release stating that they had found liquid water on Mars. Now, a number of years ago, scientists saw a series of streaks going down the slopes and mountains of Mars that appeared and disappeared seasonally with changes of temperature. Scientists predicted that these streaks could actually be liquid water flowing downhill and gave them the name Reoccurring Slope Linea, or RSLs. Now, very briefly, just like we see objects as being a certain color, so molecules have a certain, quote, color, based on the wavelengths that they absorb. And this is, of course, called the spectral fingerprint, and just like a fingerprint is unique to each person, so a spectral fingerprint is also unique to a molecule. Now, on an instrument in one of the satellites orbiting Mars, scientists were actually able to measure this spectral fingerprint in areas where RSLs were present in comparison to where they weren't, and were able to show that there was evidence for hydrated salts where these RSLs were present. Just like any other technique, certain caveats have to be taken into consideration with regards to interpreting the data. So I looked at the data, and first thing, the instrument that looks at these measurements has a resolution of 18 meters per pixel, and RSLs are 5 meters or smaller, which basically means that for every pixel analyzed, there's a lot more than just the RSL being examined. Two, the data is very noisy. A series of noise-reducing algorithms had to be implemented to pull out any sort of signal out of the noise. And three, this is where I kind of move away from looking at strictly the data itself into my own interpretation of the data. But when you look at the ideal hypothetical wavelength of what a hydrated salt would look like in comparison to what they see on Mars, they don't match up exactly. So, this is definitely water evidence that points towards water on Mars, but I wanted to look a little bit more into what they had besides this last paper. So firstly, the perchlorates, which is the salt that's supposedly hydrated, does exist on Mars, and this was directly measured by little rovers on the surface. Two, the temperature and humidity are such that liquid water can form, again, measured by a rover chilling on the surface of Mars. And three, that water in some form of ice or vapor does exist on Mars, and this has been shown in a whole bunch of different ways. Now, in combination with all this other evidence, I'd buy that RSLs are probably hydrated brine. But why the big hubbub about it? And it turns out that this year, NASA's Mars mission budget was set to be cut by Congress. And then everything in my life made sense again. NASA needs money, like the rest of the scientific world. Now, as it turns out, I put this off, getting this out for long enough that I actually know the end of the story now. Congress recently not only met the budget that NASA proposed, but actually exceeded it. So, their big old press release and everything really worked out for them. Unfortunately, for most of us scientists, we don't have anything, nearly as sexy as water on Mars to put on our give us money proposals. And unfortunately, currently, the funding climate in science is such that unless it relates to human health, it has a really hard time getting funding. This is really not a good attitude to have. So many of the revolutionary discoveries that have been made in the past 2 centuries that affect our lives every single day in huge ways is based on research that doesn't have anything to do with human health. Physicists studying electromagnetic radiation led the groundwork for TV, radio, and lasers. Scientists that was trying to find a protein in white blood cells actually ended up discovering DNA. And antibiotics was originally discovered by a scientist that was looking at the way that bacteria and mold kill each other. None of these discoveries would have been made if scientists were forced to study only things that related to human health because that was the only thing they could get money for. So, next time you're grabbing a beer casually with your congressman at the bar, be like, yo, support scientific funding. It's super cool and important. And here's a video why. So that's the download on the press on Mars recently, and of course telling the mistold and untold stories in science is the entire point of What's Up With That. Thank you very much for watching. I really appreciate it. Feel free to share this video with all your friends and all of your not friends and pretty much anyone that you run into on the street. Like me on Facebook and Twitter and stuff. Super exciting. And I'm shooting for hopefully a video a month this next year. And I have my second year exams coming up, so I'm not going to have time to do anything else with my life. So next video will probably be about my outside proposal, which I decided to do on plants. Yeah, the way you're looking at me right now is exactly what my advisor was looking at me when I told him about this. But promise it's super cool, super exciting, and yeah, look forward to that. Okay, thanks very much. I will see you next time. Bye. 40 whole minutes left.